We know now that pathogens are the cause of infectious diseases, and we've defined that word pathogen to mean any microbe that's capable of causing infection and disease in a host organism. We also need terminology to help compare pathogens to each other in terms of just how dangerous one pathogen is versus another. And that brings us to this term virulence. When you use the word virulence, you're talking about the degree of harm that a microbe can do inside the human body or the body of another organism. In other words, virulence is a measure of just how pathogenic a particular microbe is. There are some pathogens that are much more virulent and some that are much less virulent. Now, pathogens themselves contain certain molecules that we refer to as virulence factors. And these virulence factors that the pathogens produce are the molecules that help increase the virulence of a particular organism. We've talked about some of these in previous lectures. Virulence factors can be proteins, they can be other dissolved molecules within the cell, they can be the structures that surround a cell. We talked about, for example, capsules, the type of a structure that surrounds the outside of a bacterial pathogen and helps it hide from the immune system. There are also external structures called adhesins, which we didn't talk about, but adhesins are the kind of structures that allow a pathogen to physically attach itself to certain tissues. There are also secreted molecules that microbes produce that increase their virulence, that can be thought of as virulence factors. And these include things like toxins. Anytime a pathogen is capable of manufacturing and secreting a toxin, its virulence increases. That toxin is a virulence factor. Now, the reason that pathogenic microbes go to the trouble of manufacturing virulence factors is that they are able to overcome the defenses that the host may have, that it assists them in multiplying and growing, and it assists them in the kinds of processes that are helpful to the microbe but damaging to the host. Among those are things like tissue destruction. Virulence factors are both defensive and offensive. In other words, virulence factors help the microbe survive inside the host and evade the defense mechanisms that the host performs. And they are also offensive. They are the kind of things that allow microbes to survive and thrive in the host, but actually harm the host in the process. Next, let's talk about some more terminology that's related to infectious disease. These are some terms that are sometimes interchanged incorrectly and used incorrectly in general. For example, when we use this word disease, all we're talking about is a disorder in the body, a disorder in some of the structure of the body or some way that the body functions. So infectious microbes are capable of causing disease, again, because they get into the body, they grow, they reproduce, they increase in number, and they damage tissue as a result. Two terms that are often interchanged with each other incorrectly are these terms exposure and infection. Exposure simply describes this process where a host comes in contact with a microbe. Infection is different. When you use the term infection, you're implying that the microbe has penetrated into the body through those epithelial barriers we discussed and has started the process of multiplication within the host tissues. Once infection has begun inside the body, we can describe how it is proceeding by using several different terms. A lot of infectious disease is described as being either acute, subacute, or chronic. This term acute just means having a rapid onset, relatively brief period of symptoms, and a rapid resolution. So acute infections are going to last from days to weeks and are going to be rapid in their onset and, while sometimes quite severe in their symptoms, rapid in their resolution. Now compare that to the term subacute. A subacute infection is an infection that has a rapid course again, but generally has much less severe symptoms. So the term subacute translates as below acute. So it's something less than a true acute infection. Now, for an infectious disease to become chronic, it now is described as having a slow course. Symptoms of chronic infections can sometimes be severe, but are sometimes much less severe. The critical thing is that these symptoms are persisting and long-lasting. By definition, depending on the infection, the disease becomes chronic when it lasts longer than 3 to 6 months. Most sources say that an infectious disease is chronic once it has persisted longer than 6 months. Now, there's one other term I want to introduce here because it is often confused with the term subacute. That's the term subclinical. A subclinical infection is an infection where the organism, the microbe, is present in the body, but there's no signs of disease in that host. During the pandemic, we have heard about these carriers of the virus called SARS-CoV-2. Carriers can be said to have a subclinical infection, below clinical. In other words, less than what we can perceive. A true carrier has the microbe inside of them, is capable of spreading the microbe, but for whatever reason has not come down with the disease itself, have not come down with the symptoms and the disease itself. That's different from this term subacute. A person who has a subacute infection absolutely has the disease. It's just that their symptoms are less severe than others. On this slide, we're looking at a graph of the stages of infectious disease. So this graph shows us across time how an infectious disease changes in terms of what we're seeing in the host. So on the x-axis, we're looking at time going by. And remember, for an acute infection, we're talking about days to weeks for the course of the disease. You can see that the graph has been cut into columns based on how the disease changes across that time period. So if we start over here on the left, this is the time of exposure. This is the time that the organism, the microbe, is entering the body. And for the first time period here, the person has no symptoms. That's what we refer to as the incubation period. Remember, there's a difference between exposure and infection. To actually develop infection, you have to have the pathogen incubate in the body and start the processes of growth and development, growth and reproduction. The incubation period, therefore, is that time where the exposure has occurred and the infection is beginning to develop. No symptoms will be apparent during the incubation period. Next is what's referred to as the prodromal period. And this is the period of time during the development of infection when the person is beginning to feel bad, beginning to have signs and symptoms. Oftentimes, the person will describe themselves as just feeling not quite right. They're not exactly showing specific signs, but they're just not themselves. Maybe they're fatigued, maybe they have some vague muscle pains, or some other general feeling of unwellness. And that brings us into what's called the invasive phase. During the invasive phase, we have full-blown infection. And that's the time when you're going to see the typical signs and symptoms for that particular infectious disease. The invasive phase is going to reach a peak, what we call the acme of the infection. So this is when the symptoms are the worst. The infection is at its worst. Remember, during this time, the immune system is at work. Once those barriers were breached and the exposure became infection, the innate immune system was activated, and the adaptive immune system was then activated. So after the acme is reached, we go into what's called the decline phase. The symptoms are still present, but they are reducing in their severity. They're starting to be brought under control as the immune system is beating back the microbe in the body. After the decline phase comes the convalescent period. This is the time when the body is repairing itself. This is the time when the infection has been brought under control, the inflammatory response is over, and the body has now begun the process of healing, repairing any tissue damage, cleaning up any cellular debris or other types of materials that were the result of the immune assault on the pathogen. Now, for most people, the convalescent period is the last portion of infection. But some people go on to develop what are called sequelae or sequelae. And all a sequela is some kind of a remaining symptom, some kind of a problem that will persist once the infection is over, but the body cannot heal. So there are lots of different sequelae that can come about as the result of infectious disease. The virus that causes COVID-19 is showing itself to create quite a few sequelae in humans, and these are really variable. Of course, we're only just beginning to understand the coronavirus and the disease called COVID-19, but we're starting to see sequelae across all different age groups, including persistent cardiovascular inflammation, clotting problems. There are some people who are developing sort of a persistent inflammatory reaction in their nervous system. There are people who have developed paralysis weeks after recovering from the acute infection. It's really quite scary to watch as more and more people become infected with this virus. We're learning more and more about the sequelae that can develop as a result of this infection. So it's important for us to understand that there's a big difference between exposure to a pathogen, infection with a pathogen, and developing an infectious disease. It's quite possible for a person to be exposed but not become infected. It's possible for a person to become infected but not develop signs of disease. The question is then, what determines when infection and disease occur? What is it that determines why one person might develop infection but not disease, and why another person gets a full-blown case of the disease? I've listed here some of the things that help determine when infection and disease occur. Those include things like the infectious dose, any previous exposure that a person may have had to this pathogen, and of course the immune memory that comes about from previous exposure, the general health of the immune system in the person, and of course the virulence of the pathogen. Let's talk for a minute about infectious dose. There are two important measurements that we need to be familiar with regards to infectious disease dose. The first is the ID50 and the second is the LD50. The ID50 is the number of individual microbial cells or viral particles at which 50% of exposed hosts will become infected. The number of actual cells or particles that 50% of exposed hosts need to come in contact with in order to become infected. The LD50 is different. The LD50 describes the number of cells or particles at which 50% of infected hosts will go on to die from the infection. So ID is infectious dose, LD is lethal dose. And in both cases, we're looking at a population of host organisms. What is the number? What is the actual number that you need to expose people or other host organisms to so that 50% of those exposed will become infected? What is the number of cells or particles that you have to expose host organisms to so that 50% of them who've become infected will go on and die? If you look at the graph on the slide, what you're seeing across the bottom here is, on the x-axis, is the number of bacterial cells or viral particles. And what you're seeing on the y-axis is the percentage of affected people. And again, that's going to vary, that term affected, depending on if you're talking about ID50 or LD50. Affected people for the ID50 are people who've become infected. Affected people in terms of the LD50 are people who have died. So you can see that as the number of pathogenic cells and particles starts to go up, the percentage of those people who are affected also starts to go up. You see this exponential curve for a while. And then there's an inflection point where From that point on, you're going to start to see that as cell and particle number increases, percentage effective starts to drop off. You get this S curve, in other words. It's this middle point right here. It's in the center of that steep exponential curve that the ID50 and the LD50 is calculated. If you think about individual infectious diseases and the pathogens that cause them, you can really see the difference in ID50. For example, for the norovirus, it takes exposure to about 14 virions, 14 viral particles, for 50% of exposed people to become infected, to develop norovirus-associated disease. That's a tiny number of viral particles, 14. So norovirus is considered highly, highly contagious. It also happens to be highly virulent. Now compare that to influenza. Influenza is also contagious. It's just less contagious than norovirus is. It takes about 1,000 virions, 1,000 viral particles of exposure for 50% of people who get exposed to develop the disease, to get infected and develop the disease.